Okay. Well, let's go ahead and get going for today. So, announcements. Quiz 9 is next Wednesday. Is going to be due next Wednesday. So, but don't do it right now. I mean, it's released, but we need to get into chapter 9 or lecture 9 material before you be able to answer most of the questions there. Okay. So, I probably am going to hand out a problem set on Monday. Okay. As well. So I'll post that on Canvas. It'll be due a week from Monday, so the 8th. Okay. And then finally, in terms of group work, we don't have another group work assignment for a while, just so you have some idea. We probably won't do another one of those until lecture 11. So lipid metabolism. So we're ways away from that at this point in time. Just wanted to give you the heads up. We only have one more of those left in the semester. Okay. All right, so a riddle. You're standing in front of a room with one light bulb inside of it. You cannot see if it is on or off. Outside the room, there are 3 switches in the off position. You may turn the switches any way you want. You stop turning the switches, enter the room, and know which switch controls the light bulb. How do you do that? This is super important. Anyone? No one. Okay, we'll leave one of them alone. Did anyone else want to help?
Don't you have to switch 2 outer ones? If it's off, the middle one is, I guess if it's off, the middle one works. And if it's on, that's good.
There's a way to do this.
I didn't say process of elimination, go into the room as many times.
No, you can't. You can only enter the room once. No one knows? Really? No, you left the same situation. Leave the door open. Some of you are like, why do I even care about it? This is labeled. No, it's not labeled. So if you turn one on, you leave it on for 5 minutes. You turn it off. You turn another one on. You can enter the room, and if the light bulb is hot and off, then it was the one that you had on in the first place. If it's on, then it's the one you just switch on. And if it's off and cold, then it's the one you never touch. Oh, see? So you become, oh man, that was it, that's it. This just takes, I hope you're taking notes on that. That'll end up coming back to bite you if you're not careful. Okay, so on Wednesday, we had gone over these cataplerotic and anaplerotic definitions. A cataplerotic reaction is something that will reduce or pull intermediates away from a particular pathway, tending to reduce the flux of that pathway. Now we put that in the context of the citric acid cycle because the citric acid cycle this is really important for. Because the citric acid cycle is amphibolic, meaning that it can do both anabolic. It's really the starting point of many anabolic processes, but it's also the ending point of many catabolic processes. Right, and I showed here in all these different arrows, these blue guys, these would be anabolic processes that are cataplerotic reactions, meaning that they're going to lead to an overall reduction in the flux of the pathway, right? And so that means that then to maintain proper flux and steady state levels of the intermediates in the citric acid cycle, there needs to be these anaplerotic reactions. And I asked a question about this. In fact, do I have that question up? No, I don't. No, I don't have it up. Okay, so, and I don't want to go find it. But that question essentially said, which of these will lead to a net synthesis of oxaloacetate? And I said that the production of citrate from acetyl-CoA was one of the options. If you remember, the other option would be the production of alpha-ketoglutarate from glutamate, and then the activation of pyruvate carboxylase, okay, or some combination of those. So the last 2 that are actually going to lead to net synthesis of oxaloacetate and are what would be considered anaplerotic reactions for this. Now let me go ahead and show you what we mean by that. Okay, it turns out that the amino acid glutamate, really glutamine would be a good place to start. This is an N. Okay, glutamine can be converted. Let me use a different color here. There we go. Can be converted directly to glutamate by a single step. That then, by another single step, can be converted to alpha-ketoglutarate. That will feed carbons into the citric acid cycle. Those carbons then, okay, can become oxaloacetate. Right? And so essentially what you're doing here with glutamine or glutamate is you're linearizing a cyclic cycle by feeding something into a different point on the pathway. Okay, so that can lead to net synthesis of oxaloacetate that would be anaplerotic. Okay, the other aspect of this, when we come up here, we have seen this. Pyruvate can become acetyl-CoA. Of course, that takes some CO2, or that will break off a CO2. It'll make some NADH in the process. And the enzyme that we looked at that will do that is a PDC, or the pyruvate dehydrogenase complex. Okay, so that's not what we would consider anaplerotic, but we've looked at that. Now, here's what can also happen, though. Pyruvate can be converted, it can be carboxylated to oxaloacetate. Okay, you need ATP to do this, and the enzyme that does this is the enzyme pyruvate carboxylase. And pyruvate carboxylase is a really important enzyme in replacing oxaloacetate supplies in the citric acid cycle. Okay, in fact, pyruvate carboxylase, if you remember, is the first step in gluconeogenesis, but there are lots of cells outside of gluconeogenic organs, the liver and the kidney are your primary gluconeogenic organs, that express pyruvate carboxylase. And the reason is because it's so important as a producer of oxaloacetate when flux through these other pathways, these anabolic pathways, are increasing. Okay? And that will then have the capacity to then restore steady state pools of intermediates for the citric acid cycle, maintaining a proper flux of the citric acid cycle in the cell, and therefore maintaining the life of the cell. Now, let me go ahead and give you an example where this might happen. We could give lots of examples of how this might happen. But let me go ahead and give you one to just bring something that you can kind of sink your teeth into with this. Okay, so let me go to this blank slide, and we'll start here at oxaloacetate. And in the first step, of course, of the citric acid cycle, that can become citrate. And acetyl-CoA is going to be feeding into that process. Okay? Nothing new there. The enzyme there is citrate synthase that does this. The first step, and also an irreversible step in this pathway. Okay, so citrate synthase. Now, citrate synthase, okay, has to be pulled, or can be pulled, out of the mitochondria. Okay, so this would be the cytosol, to make fatty acids, or more generally, lipids. Okay? And so if signals in the cell are upregulating fatty acid synthesis, okay, you're going to see then this removal of citrate out of the mitochondria. We'll talk about that, and we'll look at the details of how that occurs, obviously, when we get to lipid metabolism, but we're not going to talk about it right now. We'll just say that it happens. Right? So citrate will start to be removed, and that means this, because we've got the rest of this pathway here. And the rest of the pathway would be essentially regenerating oxaloacetate, but if this is occurring here, then oxaloacetate concentrations will start to be depressed. Okay? Because the carbons that would replenish oxaloacetate are being used to make fatty acids. Okay? Now, if you remember citrate synthase, that enzyme, one of its primary modes of regulation is just based off of substrate availability. And oxaloacetate concentrations are going down, so that will then lead to the inhibition, okay? I'll go ahead and draw it like this. Don't think that that's allosteric, but because it's not. Oxaloacetate is not allosterically inhibiting citrate synthase, but the fact that really it's this, the fact that this is going down is going to lead to an inhibition of citrate synthase. Now the consequences of that are going to be that acetyl-CoA is going to go up, right? Because as citrate synthase flux starts to decrease, okay, acetyl-CoA, which would be one of the components to make citrate, is now going to start to go up in concentration, okay? Now, if we left it at that, that would be problematic to the cell, okay? Because all of a sudden flux through the citric acid cycle would decrease, and what the cell is saying that needs to happen to produce fatty acids, even the capacity to make fatty acids will start to go down because you can't make citrate. Right, so here's what is fascinating about this, right? This enzyme, pyruvate carboxylase, carries out this reaction that we looked at in gluconeogenesis. Pyruvate carboxylase. That enzyme is allosterically activated by rising acetyl-CoA concentrations. Okay. And pyruvate carboxylase, if you remember, is found exclusively in the matrix of the mitochondria. Right, so rising acetyl-CoA concentration will activate pyruvate carboxylase, which will then start to replace oxaloacetate, using pyruvate to do so. Okay. And that combination of things will maintain what would be considered the homeostatic stasis of the cell, or essentially the steady state of the citric acid cycle. Okay. That's one example of many that we could give. We could give lots of different things where something happens, starts to take away from something in the citric acid cycle, and then in response to that, something else will be upregulated to start to replace it. Okay? So if cataplerotic reactions start to increase in flux, then anaplerotic reactions will also have to start to increase in flux as well to maintain this steady state. Okay? Now, one more thing here. I won't use this slide to do it, but I do want to make sure that we understand this, okay? Because lipids, right, fatty acids, okay, we'll see this. Fatty acids can and will be broken down into acetyl-CoA. And that was one of the options on the question that I gave you on Wednesday. Will this lead to a net synthesis of oxaloacetate? Fatty acid breakdown to acetyl-CoA will not lead to a net synthesis of oxaloacetate, because acetyl-CoA, okay, will feed into the production of citrate, but it has to use an oxaloacetate to produce an oxaloacetate. Okay, so anything that feeds into acetyl-CoA to make citrate from oxaloacetate can't lead to the net synthesis of oxaloacetate. Okay, all right. And despite the fact that you could be breaking down massive amounts of fatty acids, it will never lead to an increase in oxaloacetate. Okay? Because every citrate that's made requires the production or the use of an oxaloacetate. So this, let me make sure I'm clear here, this is not anaplerotic. Not sure I spelt that right. Let me check. Nope. It's not an easy word to spell. There we go. Actually, I think I did get it right. Oh no, I got my R and my O mixed up. Okay, there we go. Not anaplerotic. Yeah, question?
So even though fatty acids are being broken down into acetyl-CoA, and acetyl-CoA goes and activates pyruvate carboxylase, we're not increasing oxaloacetate?
So that could happen, right? So the question would be, well, what if you break down fatty acids and then you're increasing acetyl-CoA and that then activates pyruvate carboxylase? That can lead to, okay, the increased production of oxaloacetate. The question that I asked on Wednesday said that the production of citrate from acetyl-CoA, and that can't lead to increased or net synthesis of oxaloacetate. Because every citrate that's made, you use an oxaloacetate. Yeah. Okay. And so I just used fatty acids as an example of something that could provide acetyl-CoA. Okay. You could say the same thing about glucose. Anything that provides acetyl-CoA, if it combines with oxaloacetate to make citrate, can't be antiproduct. Yeah. Okay. Other questions? Okay, so this is one example of many that we could give, okay, and that's probably one of the more important aspects of the citric acid cycle in terms of trying to understand the metabolism as a whole about that cycle, okay. Now, what I want to spend the rest of, well, not the rest of the day, but most of the rest of the day talking about is one of these special topics, and I want to turn to cancer metabolism. So for a number of reasons, but obviously probably one of the more important reasons is just the importance of cancer in terms of overall health, right? This is a way people die, and in fact it's a way a lot of people die, okay? So it's something that literally plagues us as a human race. And cancer is a really interesting study in metabolism because cancer cells have essentially reprogrammed metabolic output. Okay, they do things that what it would be considered a quiescent cell would not really do. Okay, by quiescent cell, I simply mean a cell that's not undergoing active division. Okay. Cancer cells would be the opposite of a quiescent cell. These are not only undergoing active division, but they are undergoing uncontrolled active division. Okay, that would be the definition of cancer. And if you think about that from a metabolic standpoint, just from a cellular standpoint, these cells have to double their biomass every 24 hours. Okay, mammalian cells. A mammalian cell doubling time is about 24 hours. And in that 24-hour period of time, that cell then has to double the amount of lipids it has to double the amount of DNA it has to double the amount of protein it has, right? So anabolic processes are going to be really ramped up in these cells, okay? Now, because of that, then these cells also have to consume a significant amount of energy to maintain the energy needed to run those anabolic processes. Not only do they have to consume a lot of energy, but they also have to have just the building blocks to build the nucleotides and the lipids and the amino acids that they will need to continue cellular division. Okay? So to do that, cancer cells do some really actually, well, from a scientific standpoint, some interesting things to metabolic pathways. Okay. And so one of the things that we want to look at, there's actually a couple of things that we want to look at. We'll make our connection to the citric acid cycle here in a moment. But one of those things that we want to think about is the Warburg effect. Okay, the Warburg effect, many of you have probably heard of the Warburg effect. This was discovered by Otto Warburg in the 1930s. We've known about this for almost a century, that cancer cells absorb significantly more glucose than your normal quiescent cell. Not only that, but they will also spit out significantly more lactate than your typical cell will do. Okay? In fact, cancer cells will increase utilization of glucose 20-fold, okay, relative to a quiescent cell. So they'll suck down glucose at a 20-fold higher rate than your typical cell. Okay. Now, interestingly, and like I said, in terms of the Warburg effect, 85 to 90% of the glucose that makes it to pyruvate will be converted to lactate. Okay. So 85 to 90% of that makes it to pyruvate will become lactate. And they will then excrete significant amounts of lactate, okay, these cells. In fact, in culture, cancer cells will decrease pH of the culture medium very rapidly because of the amount of lactate that they'll spit out. Okay. And they will do that, interestingly, even in the presence of oxygen. Okay. So they don't have to go anaerobic to do this. They'll do it even in the presence of oxygen. Okay. Now, a couple of things to say here. When I say cancer cell, I'm using that as a very general term. Okay, and we're talking about this in terms of general phenotypes of cancer cells. You start to look into literature of cancer, and you're obviously going to find that there are lots of specific types of cancer. Each of those are going to have their own phenotypes. Some of them would depart from this. Okay, so this is a general description of cancer as a whole. Okay, now what's also interesting about this is that embryonic tissues, healthy tissues in an embryo, do essentially the same thing. Okay, in fact, there are lots of similarities between embryonic tissues and cancer tissue. Okay, it's, I mean, there are some notable differences, but there are definitely a lot of similarities. Okay. Now, in terms of this, right, glucose metabolism, increasing absorption of glucose, and then making lactate, there's really two questions. How? How do they do that, and why do they do that? Okay. Well, we can give some very good answers for how. The why is still, we've still got some good theories out there, but you know, it's not entirely understood in all cases. But let's talk about the how first, okay, because we can give good answers. One of the aspects of cancer cells is that they will express high levels of hexokinase 2 and GLUT1. Both of these have really low KMs, which means that they can obtain fairly rapid rates of catalysis at fairly low concentrations of glucose. Okay. Expression of GLUT1 being high in the membrane, that means that it has pretty massive ability to absorb glucose. High expression of hexokinase 2 means that if it does get into the cell, it can be trapped almost immediately. Okay. So, so that's essentially some of the how this happens. Okay, glucose gets into these cells. Now, we are talking about, in this regard, on this figure here, we're talking about this here, and we're talking about this step right here. Okay. Now, the other aspect of all of this, okay, because hexokinase 2 can be inhibited by glucose 6-phosphate. However, these cells, okay, will have a high level of AMP because anabolic processes are so ramped up, so they'll have lots of AMP. That will activate PFK1, as we've talked about. So this is PFK1, which means flux through glycolysis and utilization of glucose 6-phosphate is high. So even though they're bringing it in and trapping a whole bunch of glucose, it's being used immediately as well, so they never get the feedback inhibition of stop absorbing glucose. And that's why they'll just keep sucking it in. Okay. Now here's what's really interesting about this as well. Okay, again, from a scientific standpoint. This here, PKM2, on this figure, this is pyruvate kinase. You all say to yourself, doesn't that catalyze the last step in glycolysis? Exactly. And it's irreversible, right? Exactly. Very good. Pyruvate kinase in cancer cells, many cancer cell types, will express a form of pyruvate kinase that is not higher in activity, it's actually low in activity. Okay, and that's what's being set up here. Activity is down. So this is depressed activity. And you think, well, that doesn't seem right. Hexokinase is high in activity, PFK1 is high in activity, and then the end of it is low in activity. Well, what that does is this. It produces a bottleneck. And it produces a bottleneck at the very end of the pathway. So you have huge flux in the beginning of the pathway, and then at the end of the pathway, it narrows down in terms of flux, so there's not as many carbons leaving the pathway. And what does that do to concentrations of intermediates in the pathway? They go up, right? And so what happens is steady state levels of glycolytic intermediates go up. Why is that important for these cells? Well, because these intermediates, okay, are all used in pathways that will lead to nucleotide synthesis and amino acid synthesis. But nucleotide synthesis is very important. 3 billion bases have to be replicated every 24 hours. Right? That's a lot of work for the cell to do. Okay, so those, right, increasing, okay, concentrations of these intermediates is going to lead to massive amounts of nucleotide synthesis. Okay, and this pyruvate kinase providing this bottleneck is very important. Now, any pyruvate, not all of it, but again, 85 to 90% of it that slips through here, okay, lactate dehydrogenase will make most of that in the lactate. Okay, if you remember, okay, this is going to recycle in ADH. And one of the theories, this is a very simple theory, why does most of that pyruvate get converted to lactate? Well, it might be just simply to replace NAD+ supplies to maintain high flux up here. Okay. And that's a theory that I'm willing to go along with. We have to, you know, I mean, these theories have to be supported though by data. So, and there's some conflicting data on that in that regard. Okay. Now, what's also interesting about this is that that means that not a lot of this pyruvate is getting into then the mitochondria and being used in the process of the citric acid cycle. Okay, and we'll talk about that in our next slide. Yeah, but that would be the implications of this. Okay. Questions on this? Yes?
When you were talking about earlier, you were talking about embryonic stem cells.
Mm-hmm.
Oh, are these stem cells or are they something else?
Well, certainly. So I would have to go and look real carefully if the metabolism of what I mentioned, if it was stem cells. My hunch is that it is the embryonic stem cells. I mean, especially within, because what I'm referring to really is the very initial stages of development, like 4 to 6 rounds of cellular division, and all of those are stem cells for sure. Okay, and those cells have metabolism that looks very similar to this.
Okay, so they're like very beginning.
Yep. Okay. Yeah.
Are there any therapeutics that currently target this?
Okay, yeah, so the question is, are there currently any therapeutics that would target this? So that's an interesting question. Over the last decade, or even 10 to 15 years, there's been a change in the focus of the development of therapeutics from, targeting cell cycle regulation and genetic expression to targeting metabolism. And lactate dehydrogenase at one point in time was a pretty promising, would seem to be a pretty promising drug target for some cancer cells, though it's not really panning out because it's hard to develop a drug for it that's real specific. Okay, but I can say this: you can knock down expression of lactate dehydrogenase in many cancer cells, and they'll die. Okay, so that's fairly interesting. Okay, from that standpoint, I can say this even on this slide alone. Okay, metformin, which we said can be a treatment of diabetes. Okay, well, there was this meta-analysis study that was done on people with type 2 diabetes, and their mortality rate for cancer was significantly lower when they were taking metformin across the population. And people thought, well, that's kind of strange. Why is that? And it turns out that metformin activates AMP kinase, and AMP kinase inhibits nucleotide synthesis. Okay, so metformin is considered by some, and there's still, again, conflicting data on this, so we don't have like the full-on answer to this, but metformin is thought to be a potential anti-cancer therapeutic, okay, with fairly small side effects, actually. I mean, relative to some, chemotherapies. It's not as significant in that regard, okay. So, but it can activate AMP kinase, and AMP kinase then, okay, inhibits this. We haven't talked about mechanisms that AMP kinase will do that, but it certainly will inhibit this. Okay, but so I mean, to answer your question, yeah, I mean, there's thought to that, right? I mean, how can we develop anti-cancer therapeutics at this level. Problem is, targeting glycolysis in the citric acid cycle is problematic, right? So, from a side effects issue. I mean, cyanide's great, anti-cancer therapeutic, but it kills some other things along the way. Okay. Questions? Yeah. I think that, so the LKB1, I'm not going to talk about that. That was just part of this figure. I think it stands for limb kinase, but I can't remember. That's me guessing. Total guess in the dark. Yeah. I do know this. The Rockies won on yesterday, if you were wondering. It was, that's not a guess in the dark. It was 6-3. It was a good game for us. They play again tonight, if you're really curious. So. And for those of you, I know there was some concern amongst the class that I was not wearing the vest. It was bothering some people. Some people were like kind of hyperventilating about it. It's okay though. I brought it back, but I can't promise I'm going to wear it for the rest of the semester. So just keep that in mind. Okay. All right. Now let's do this. Let's go one step further because we want to connect this to the citric acid cycle, in particular anaplerotic and cataplerotic processes. We don't have a lot of, in cancer cells, generally speaking, we do not have a lot of pyruvate feeding into the pathway and into acetyl-CoA. Okay, that's one of the aspects of this. The other aspect of this, and is very similar to this slide that I was drawing here, okay, there is a lot of lipobiogenesis. So in a cancer cell, that's definitely happening. Okay, so we come back here, okay, and what you have on this slide, let me just show you this. You have the citric acid cycle, and that step that I just highlighted on that last slide is this guy right here. Okay? Citrate is being pulled away to do lipobiogenesis by these cells at a fairly rapid rate. Okay? Not only that, but there's a significant amount of this going on. Okay? Actually, let me do it like this. Significant amount of this here, where oxaloacetate is being converted to malate, shipped out of the cell, and then this enzyme here, ME, that's very nonspecific and vague, but it stands for malic enzyme. That enzyme converts malate to pyruvate and makes NADPH. And you think to yourself, isn't it NADH? No, it's NADPH. Okay, NADPH is a coenzyme that's virtually identical to NADH, except for it has a phosphate group on one of its sugar rings. Right? NADPH is a really important coenzyme in biogenesis pathways. Okay, lipid metabolism, for instance. Okay, we don't have an appreciation for this because we haven't really gone over any of those pathways. Right, but these cells, not only do they have to have a lot of carbon skeletons to make lipids or to make nucleotides or to make these things, but they also have to have reductive power. Okay, and NADPH supplies that, and this is one of the more important steps in developing that NADPH. Okay, but to do that, you need malate, and to get malate, you need oxaloacetate. So these cells have this significant draw on oxaloacetate, which will then reduce flux through the citric acid cycle, okay, because of these two what would be considered cataplerotic processes. Okay. Now, interestingly, okay, and we've known this for like 50 or 60 years, okay, many cancer cells are essentially dependent upon the amino acid glutamine. Okay. They will absorb significantly more glutamine than most other cells and most other amino acids. Now, why? Okay, well, glutamine, okay, and I'm just going to draw this all the way through. Glutamine, when it gets absorbed in the cells, can be transported into the mitochondria and converted to alpha-ketoglutarate. And this is the more important, or one of the more important anaplerotic processes for these cells, because they will take alpha-ketoglutarate, feed that in the citric acid cycle, and restore oxaloacetate concentration. Now you say to yourself, well, why not pyruvate carboxylase? Like you did on our last slide, right? 2 or 3 slides ago, I said, well, you just, acetyl A concentration goes up, and that activates pyruvate carboxylase, and that then makes oxaloacetate. Why don't these cells do that? Well, there's 2 problems with that. First of all, most of the pyruvate is getting converted to lactate. You don't have a lot of it going into the mitochondria to do this. And the other problem with that is that a lot of these cells don't have pyruvate carboxylase, or they have a very small amount of it. Okay? So glutamine becomes more and more important in this. Now, the other aspect of this, okay, is that as glutamine gets brought into the cell and is converted to glutamate, and then alpha-ketoglutarate, I like this figure. They draw a little red box. This is what they have down there. If you can't see that, it's like this. Okay, and that's really, really helpful. That's nitrogen. Okay? All right, so glutamine, if you remember, and it's an amino acid that has 2 nitrogens on it. It has an alpha amino group, of course, okay? And it has then also on its side chain, it has a nitrogen, okay? So nitrogen is really important to make nucleotides. You have to have nitrogen to make nucleotide bases, okay? So, and you can see that up here as well, right? Right, these, right, pyrimidines, purines, that kind of thing. There's processes outside of the mitochondria that are going to make these as well, okay? So all of this is going to lead to alpha-ketoglutarate. So glutamine is very, very, very important for these cell types as well, okay? Again, painting with a very broad brush, you can find cancer cells that don't need glutamine to survive, okay? But you also will find significant a significant phenotype in many cancer cells that require a glutamine in culture, okay, or in their surroundings. Yes?
So are these products of this changed metabolism also harmful to the body, or is it mostly just the extreme growth of the cell?
Okay, yeah, so the question is, would the change metabolism of this be harmful to the body, meaning what they're going to start to excrete, okay? Or is it really just the harvesting of the available energy? It's going to probably be more the latter than the former, okay? And that is just the available resources that are being used up in a more rapid way, okay? One of the things that we'll build a deeper appreciation for when we get to amino acid metabolism is that glutamine is one of the primary ways that the body transports nitrogen to the liver, okay? And it's the way that it gets rid of nitrogen. Nitrogen is a waste product that can be, if it accumulates, can be very toxic. So there's a lot of glutamine in the blood, okay, that these cells will start to absorb. Of course, they're absorbing lots of glucose. Okay. And that can then upset homeostasis of surrounding tissues. And if, surrounding tissues is the brain, that's a bad deal, right? Or if it goes metastatic, that can be really, obviously harmful. Yes?
Is there any other type of cell, whether it's, I'm not sure, I guess, normal or diseased or mutated in some way other than cancer cells? Is there any other type of cell that uses glutamine like this?
Well, not to this extent. Yeah, so the question is, would there be any other cell, normal or otherwise, that we use glutamine to the other extent? I couldn't speak to other disease states besides cancer. There may be other disease states that would, some cells that would require more glutamine. Normal, though, healthy tissues, typically no. I'm not sure if embryonic stem cells use that much glutamine either. I don't know the answer to that. My suspicion is that yes, they do. They certainly use the glucose. Not sure about the glutamine. Okay. Other questions? Yeah?
So like with decreased TCA cycle activity, like I know that glucose obviously, or like glycolysis has substrate level phosphorylation of DHT,
Yeah.
but like how are they getting their energy?
Yeah, okay, so the question is how are they getting their energy? because you have a depressed citric acid cycle. You don't actually, because this can actually increase energy production through the citric acid cycle. Okay, so these can actually still sustain energy producing pathways through oxidative phosphorylation, but they use alpha-ketoglutarate or glutamine instead. Okay, but the other aspect of it is that they do produce just a significant amount of their energy needs through glycolysis. Okay, it's not as efficient, clearly, but they don't care. They just suck down more glucose. Right? So, cancer, nasty stuff. Question? Other questions? I think it is important, one more thing to just recognize, okay, many of the enzymes that are listed here, and we'll get to them when we get to amino acid metabolism, first of all, are anti-cancer therapeutic targets, okay, because of their role in maintaining cancer cell health. The other aspect of these is that a lot of them are not really mutated. They just undergo increased expression, okay. So genetic expression, of course, changes for cancer cells as well. You have increased expression of glutamine transporters, increased expression of enzymes that carry out glutamine metabolism too. Okay, I think that that's it for cancer for now. We will actually come back and talk a little bit more about cancer and make some more connections as we go on throughout the class. Okay, but let's go ahead and do this. Let's switch topics here. And let's go ahead and move to oxidative phosphorylation, okay? So that kind of leaves behind then the citric acid cycle, and we enter into oxidative phosphorylation. This is a fairly long set of slides. Lots to cover here. We'll start at it from a really broad standpoint, just chemiosmotic theory. What do we mean by that? What is that? Moving into then electron transport chain, we'll talk about something called PO ratios, okay, very important aspect in terms of how much ATP we can get per NADH and FADH2. And then finally we'll get down here to structure and mechanism of ATP synthase, which is pretty fun stuff. Okay, lots of interesting stuff on this, but let's start here real broad. The chemiosmotic model or chemiosmotic theory. We'll be thinking about this from a mammalian standpoint, but certainly chemiosmotic theory and model would be something that would be broadly applicable to most biological organisms. And we'll describe more than just oxidative phosphorylation. It describes lots of different processes in a cell. Certainly photosynthesis uses, photosynthesis would be another example of the chemiosmotic theory at work, okay? And that would just be one example of many. Okay. Bacterial cells do this as well. Okay, so obviously, well, I don't know. The drawing's not great, so maybe this isn't obvious. But that's supposed to be the matrix, not the matrix. It's supposed to be the mitochondria. Okay, double membraned organelle. The outer membrane, just for our purposes and just so we understand this, and I've already mentioned this in this class, but the outer membrane is freely permeable to small molecules. Remember, it's got these big porins in it. Okay. So it lets things fly in and out. Cytosolic environments are very similar to inner membrane space environments. Okay, the inner membrane is definitely not soluble. Okay, so the inner membrane is impermeable to most small molecules, and it comes equipped with significant amounts of protein. Okay. In fact, about 90% of the membrane is protein, and only about 10% lipid. So it's really more protein stuck together than it is lipid stuck together. Okay, so lots and lots of proteins in it. It has what we will call the electron transport chain or respiratory chain. Okay, ATP translocase, we'll talk a little bit about, ATP synthase, and then other membrane transporters. Okay. These are the two that we'll talk about, though, in this upcoming set of slides. Okay. And then finally, of course, the matrix of the mitochondria, that's where pyruvate dehydrogenase complex is found, citric acid cycle is found. When we get to fatty acid beta oxidation, that's going to be found there. We'll talk about amino acid oxidation. Those are found there, and then a whole bunch of other stuff. Okay, very densely packed environment. Okay. Big thing for us, though, would be to remember that there is the inner membrane, and then there's the space between the two membranes called the intermembrane space, and then, of course, outside of that is a cytosol. Okay, and inside of that's the matrix. Okay. Now, in that context, then we can start to think about the chemiosmotic theory or model, and this would be you know, what might be considered the 5,280-foot view of the chemiosmotics model. Okay, looking at this from a very broad standpoint. Okay, we would have here, in here, you could call this the matrix. Okay, we will also call this, though, the inside. Okay, importantly. In standing for negative. Okay, and that would be based off of the local charge around the membrane. I'll just circle some of this. So in for negative. Okay. That would make the exterior of this out here, this would be called the P side. Okay, or if you're talking about if you are talking about a mitochondria, then it's the intermembrane space. Okay. But that would be our membrane potential setup. It's positive on the outside, negative on the inside. Okay. Now embedded in that, again, this is 5,280-foot view of this, but embedded in that will be something that we call the electron transport chain, or respiratory chain, comprised of complexes, complex 1, 2, 3, and 4. We'll look at them in detail, but those complexes as a total will take electrons from reduced electron carriers, this would be NADH, okay, primarily would be the diffusible one, pass them through a series of reactions to oxygen, oxygen being the final electron acceptor, and make water in the process. That's very exergonic. And we'll look at numbers here. I don't think we'll get to the numbers today, but we'll look at numbers. Okay? It's exergonic, and that energy is going to be used to transport protons out of the inside into the P side. It establishes a proton gradient and maintains the proton gradient. Okay? Those protons then, you'll see out here, they're trying to illustrate that there's lots of protons out here. Okay? Those protons will then fly back through the membrane through a molecule commonly called ATP synthase or complex 5, and that will be, that energy as it goes back with its gradient will be used to make a lot of ATP. Okay. And the bulk of the ATP for the cell is made through this process. Okay. Our job over the next several days, okay, is going to be to look at all those complexes, to look at this, okay, and talk about this from a thermodynamic standpoint as well and a mechanistic standpoint. How do they do this? Okay. Isn't that exciting? I'm excited about it. But for now, we're going to stop because the Rockies start in 2 hours. So we got to go, you know, we got to figure out what to do the next 2 hours before we start watching the Rockies play. Okay. All right. Have a good rest of your day and a good rest of your weekend, okay? Or a good weekend. What's up, man?
Help me understand something.
Okay.